Hello, you're watching At the Bench with Norgen Biotek. Today, we will provide a tutorial on a step-by-step workflow for the Urine Exosomes Purification and RNA Isolation Minikit, product number 58400. To avoid contamination of samples, sterilization of your work area and instruments using 1% bleach, followed by wiping down with 70% ethanol is recommended prior to starting. Additionally, preparing a biohazard bag for the removal of wastes such as flow-through and associated tips, tubes, and other wastes is recommended in compliance with health and safety regulations. In this kit, you will find a detailed product insert, collection tubes, 1.7 milliliter elution tubes, mini filter spin columns, mini spin columns, XOR buffer, wash solution A, Lysis Buffer A, slurry E, Lysis additive B, Exosome Buffer, elution solution A. It's highly recommended to warm up Lysis Buffer A at 60 degrees Celsius for 20 minutes, and mix well until the solutions become clear again if precipitates are present. Prepare a working concentration of wash solution A by adding 42 milliliters of 96 to 100% ethanol to the supplied bottle containing 18 milliliters of concentrated wash solution A. This will give a final volume of 60 milliliters. The label on the bottle has a box that may be checked to indicate that the ethanol has been added. Collect and transfer 15 to 50 milliliters of the urine into a conical tube and centrifuge at 200 G or 1000 RPM for 10 minutes to remove urine exfoliated cells and debris. Decant cell-free urine into a new 15 to 50 milliliter conical tube. Centrifuge the cell-free urine at 1800 G or 3000 RPM for 10 minutes to remove any residual debris or bacterial cells. Transfer cell-free urine into a fresh 15 to 50 milliliter conical tube. To 1 milliliter of urine, add 100 microliters of Exosome Buffer, followed by the addition of 200 microliters of slurry E. Take note to mix slurry E well prior to use. For optimal performance, ensure that the resin is completely resuspended. Mix well by vortexing for 10 seconds. Let stand at room temperature for 5 minutes. Mix well by vortexing for 10 seconds. Centrifuge for 2 minutes at 2000 RPM. Discard the supernatant. Apply 200 microliters of XOR buffer to the slurry pellet and mix well by vortexing for 10 seconds. Incubate the slurry pellet resuspended in 200 microliters of XOR buffer at room temperature for 5 minutes. After incubation, mix well by vortexing for 10 seconds. Then, centrifuge for 2 minutes at 500 RPM. Transfer the supernatant to a mini filter spin column assembled in an elution tube. And centrifuge for 1 minute at 6000 RPM. Do not discard the flow-through, which contains your purified exosomes. Your exosomes are now ready for RNA isolation or any other downstream applications. Add 300 microliters of Lysis Buffer A and 37.5 microliters of Lysis additive B to the 200 microliter XOR buffer containing the purified exosomes. Mix well by vortexing for 10 seconds. Then, incubate at room temperature for 10 minutes. After incubation, add 500 microliters of 96 to 100% ethanol to the mixture from step 2. Mix well by vortexing for 10 seconds. Transfer 500 microliters of the mixture from step 3 into a mini spin column. Centrifuge for 1 minute at 3300 G or 6000 RPM. Discard the flow-through and reassemble the spin column with its collection tube. Repeat step 4 one more time to transfer the remaining mixture from step 3 into the mini spin column. Apply 600 microliters of wash solution A to the column and centrifuge for 30 seconds at 3300 G or 6000 RPM. Discard the flow-through and reassemble the spin column with its collection tube. Repeat step 6 one more time for a total of 2 washes. Spin the column empty for 1 minute at 13,000 G or 14,000 RPM. Discard the collection tube. Transfer the spin column to a fresh 1.7 milliliter elution tube. Apply 50 microliters of elution solution A to the column and centrifuge for 1 minute at 2,000 RPM. Followed by 2 minutes at 8,000 RPM. For maximum recovery, transfer the eluted buffer back into the column and let stand at room temperature for 2 minutes. Centrifuge for 1 minute at 400 G or 2000 RPM, followed by 2 minutes at 5800 G or 8000 RPM. Exosomal RNA is now ready for downstream applications. Thank you for watching this tutorial. If you enjoyed the video, please like and subscribe, and remember to check us out on all of our social media platforms. We'll see you next time at the bench.